So our next speaker is over there. He is a senior. He is studying chemistry, but I don't like to think of him as a chemist. I think of him as an engineer. But the building blocks of his engineering are far smaller than what any of us can see with our naked eyes. As a matter of fact, no human has ever been able to see, without the aid of a device, what his building blocks of engineering are. Yet, we have miles and miles and miles and miles and miles of this very building block within each and every one of us. It is what produced us. It is what produced the first living organism. Yes, you can probably guess what it is, but I won't tell you because you'll figure it out. He wants to bring to light something that will change our world, is currently changing our world, and has a little bit of a negative image in a lot of people's eyes. So he wants to set the record straight and show you some of the amazing things that this kind of engineering can do. So without any further ado, Mr. Petar Todorov.
Hello everyone. I'm here today to talk to you about synthetic biology. And I'll just pass this envelope around. Don't open it until I say so, just pass it around. So what is synthetic biology? I bet most of you have never even heard of this. Synthetic biology is the modular design of new biological parts and systems for a useful purpose. And here I've shown a piece of DNA called a plasmid. Bacteria carry these around, and they have genes on them. And in the last 50 or so years, scientists have figured out how to cut and paste these genes, make that bacteria smell like pineapples, detect explosives, or give you a full color palette to paint with. And the underpinning of synthetic biology is to actually take these parts, standardize them, make them plug and play, and make them available to everyone. Allow people to sample genetic material and build with it, so we don't all have to reinvent the wheel. Now, unfortunately, genetic modification has the connotation of agribusiness, big crops, GMOs, and that debate has hijacked the discussion about genetic modification in synthetic biology. It's actually much bigger than that, and it's unfortunate that we keep focusing on this. I'm not going to ask you to think bigger. I'd like you to think smaller. Actually, on the scale of the yeast that's making these bubbles. We've had a 7,000-year relationship with this organism, ever since our ancestors had their first beer. And it turns out it's still relevant and useful today. We can use it to tackle challenges that still plague humanity, like malaria, a disease that affects over 200 million people, and every year 600,000 die. We can cure malaria, but the problem is that the parasite that causes it has started developing a resistance to the drugs we use. One of our last lines of defense is artemisinin, which is found in sweet wormwood, Artemisia annua. The problem with this crop is that it takes 15 months to produce. It's susceptible to drought, pests. Sometimes you can't get a harvest out. So a team of scientists in California thought about this and plucked the genes to make artemisinin from this organism and put them into yeast. Now, it takes 3 months to make the drug. That's 5 times faster. And instead of a farm, the factory that produces it looks more like a local brewery. And there's other challenges that yeast can help us tackle. The same group decided to apply this to fossil fuels. That's a touchy subject. We're dependent on fossil fuels, and we know we have to change, but the alternatives aren't there yet. There's too few of them. Turns out yeast offers another option. It's versatile. It can make biodiesel just as well as it can make drugs. And here, it's making an organic lipid that can turn into a lubricant or a fragrance. And all of that is enabled by the systematic and modular design of life. The design of life. Let that sink in for a second. In 2010, the John Craig Venter Institute looked at this organism, Mycoplasma mycoides. It's a small bacteria with an even smaller genome. And they took that genome, they downloaded it from the internet, and they put the data into a machine that looks like this. They printed that genome, assembled it, put it inside of the bacteria, and it booted up. Essentially, they had changed one of the motors of life. They had created an entirely synthetic genetic code. This is a great step for man, because now we can code in DNA. The idea of interchangeable parts comes closer to fruition. And maybe one day, instead of writing apps for phones, we'll code in DNA for life. Maybe we'll make polka-dotted roses to give to our significant others. But we shouldn't lose track of the ethics behind this issue, as it becomes easier to put DNA together like Lego bricks. What we have to realize is that what we create won't stand alone. It will divide, it will multiply, and maybe it will take a life of its own. And that's not something that has ever been a problem before in engineering. Luckily, the Presidential Commission for Bioethics has examined this issue, and at the time, in 2010, they actually concluded that the regulations in place for genetic modification are sufficient for synthetic biology. That may not be the case in the future, as gene synthesis becomes more commonplace. And in order to have an informed debate about security, about bioethics, our population needs to be educated. And education is not going so well. Although our scientific literacy has been rising for the last 25 years, it's a fact that nearly half of Americans don't believe that non-GMO tomatoes have genes. It's a little embarrassing. But there's still hope to change from an emotional response to an informed one. And if we have to talk about emotion, we should talk about fear. Is this dangerous? Does it pose a threat to our security? And whoever has the envelope right now can open it up and take a look inside. I assure you it's safe. For everybody else, what they'll be looking at is this. This is the sequence to Ebola virus, Zaire strain from 1976. It's a virus that kills about 90% of the people it infects. And it's available online, on a government website no less, with no restrictions. Anybody can log on to it and pull this data. So why is that? Why is that when a virus like this is currently rampaging through West Africa, and the World Health Organization has called this one of the most challenging outbreaks yet? That's because the code itself is harmless. The code itself would need to be built, and only a select few companies can do that. They collaborate with the FBI, with other scientists, and they screen for sequences like this. That's not an issue yet. The issue that could arise currently in synthetic biology is an accidental release of something much less harmful, like an antibiotic resistance gene. But even then, we have to take into account that the actors in this space aren't motivated by evil. They're motivated by good. They want to have fun, but they're still bound by legal and ethical guidelines. In fact, our synthetic biology team here at Tufts has to submit a form to the University Biosafety Board before doing any of its projects. And our ideas, again, are there to improve the world. We want to fight biofilms, make cheaper antibodies in order to detect cancer or treat it, and even make antivenoms without ever going near a deadly snake. But we're only a small portion of the synthetic biology landscape. We're a blip on the radar. There's even a commercial component of this. None of this would likely ever come into your homes. But last year, 2 scientists ran a Kickstarter crowdfunding campaign to create a glowing plant. And they secured that funding. They're actually shipping the seeds this September. I'm going to get a few of them. But I bet no one in this room even heard about this. And yet, it's coming into the world. It's being released. So, unlike the other speakers tonight, I don't want to give you any homework. But I do want to leave you on this note. Synthetic biology is no longer state-of-the-art. It's no longer limited to the university labs that it happens in. And it's going to revolutionize our health, our industry, and our everyday life. So, get ready.